Welcome. It is Mr. W, and I am so glad to be here with you. Tonight's show, we're going to talk about membranes. Membranes are unbelievably important. They're an incredibly important part of your success in your biology course. And membranes are an 8-nanometer barrier between order and chaos. Membranes are a selectively permeable nano-bouncer, deciding what can enter the cell and what can't, making decisions about who can get in, who can get out, and who is either stuck inside or left on the outside. Membranes are dynamic and fluid. You can see them moving around here. All of those pieces are moving around. Membranes are built from phospholipids, these molecules over here, and they organize themselves into a phospholipid bilayer. That's an incredibly cool process that we'll be looking at today, how that self-organization happens. Membranes, by weight and function, are dominated by proteins. We'll see that phospholipids form the framework, but it's proteins that are doing most of the interesting things that are associated with membranes. Yeah, that's right. That's what we're talking about tonight. We're talking about membranes. Welcome to the AP Bio Show, where it's all about your success in your AP, IB, or college biology class. And I am so happy to have you here with me tonight. We have a special topic, sickle cell anemia, and that's because this month is National Sickle Cell Awareness Month. It's an incredibly great illustrative example of the topics that are in AP biology, and it's important to know about because it's one of the most important genetic diseases on this planet. There will be a weekly quiz. As always, there's going to be time for your questions and tips for learning the greatest of all subjects, biology. I am, again, so happy to have you with me. And if you'd be bold enough just to put your name into the chat, tell me where you're listening from, that would be really awesome because, again, this show is all about you. Just to introduce me a little bit to you, my name is Glenn Wolkenfeld, also known as Mr. W. I'm a retired AP Bio teacher. I love teaching AP Bio, but I've moved to teaching it this way. I was known as Mr. W to my students, and you can call me that too, because now you are my student, now that I'm out of the classroom and teaching through YouTube and through other means that I'm about to tell you about. And our mission here at learn-biology.com and the Science Music Videos channel is to help you succeed in AP, IB, or college biology. One of the main ways that we're going to help you succeed is through the learn-biology.com website. It's a College Board approved website that basically teaches you everything you need to know to succeed in biology. I really hope that you sign up. You can sign up with a free trial and try it out. I'll tell you more about the features a little bit later in the show. Let's go on and let's start learning about cell membrane structure. And I want to thank those of you who have already commented in telling me where you're from. We've got guests from Georgia tonight, the great state of Georgia. So thank you very much for joining us. And I'm very interested in hearing where other folks are from as well. So cell membrane structure, that's topic 2.4 of AP biology. Let's start by talking about the function of the cell membrane. It separates the highly organized contents of the cell from the cell's environment. Life is all about creating order. Outside of the order of life is the relative chaos of the environment. And life is about creating order. Membrane separates that order from the entropy and the chaos outside. The key term connected with cell membranes is selective permeability. Membranes are selectively permeable. They allow the passage of some substances and particles through, but not others. Membranes are picky. Membranes are like a bouncer. They'll let certain things in, but not others. Selective permeability looks like this. Here you see one type of molecule that's being allowed to go through. This can make it right through the phospholipid bilayer. Here you see another molecule that has to make it through a channel, but nevertheless, it's allowed to go through. We've got a third molecule that's getting pumped into the cell through the use of energy. But this molecule, which looks like some protein over here, can't make it through. It hits the membrane, it bounces off. And this substance on the inside, which is glucose 6-phosphate, that's an early molecule that's created during cellular respiration, that's not allowed out because that's an energetic molecule that the cell is going to metabolize so that you can run and jump and think and do all the things that you do in life. So cells are going to keep the good stuff in, they're going to keep the bad stuff out, they're selectively permeable. The key molecule to understand in terms of understanding membrane structure are phospholipids. They're the key structural components. They've got a hydrophobic non-polar tail. Non-polar, that means it doesn't dissolve in water. It's hydrophobic, it's water-fearing. There's a hydrophilic polar head. Hydrophilic, water-loving, dissolves in water. Water itself is a polar molecule. Hydrophilic and polar things also dissolve in water. And they're held together by a molecule of glycerol. That's at number 2 over here. Here's a cartoon rendition of the same that's widely used, something like this to represent membranes. In water, phospholipids form bilayers, bi-2, so 2-layered structure. So you should imagine this as a kind of slice through a bubble of membrane. And what's amazing is that cells are composed of cell membranes. Cells are bordered by a cell membrane, but these molecules, these phospholipids, because of their structure, hydrophilic head, hydrophobic tails, will spontaneously form this structure. And that is super important in terms of understanding the origin of life. When we're trying to explain where cells came from, there were non-living abiotic processes that enabled little bubbles of membrane-like stuff to form. And that began the process of segregating life from non-life. Here's something to know. In solution, the heads bond with water, while the tails form a water-free zone. So this over here, that's a water-free zone. This over here, a little bit larger, that's a water-free zone. That creates a bilayer, this 2-layer structure. One layer, the second layer. And the phospholipid bilayer itself is stabilized by weak bonds, van der Waals force bonds, that form between these hydrophobic tails. So there's a kind of force that's holding them together, plus this orientation of the hydrophilic heads and the hydrophobic tails. Here is an animation from my video membranes that shows what happens when you mix phospholipids with water.
Because when phospholipids into water get submerged, a phospholipid bilayer structure will emerge. The tails hang together in a water-free zone. Hear the hydrophobic moan, water leave me alone. While the heads are sticking out, touching all those H2Os. Tails in, heads out. It's how every membrane goes. Tails in, heads out in a cellular sphere. It's the bilayered basis of membranes everywhere.
The key idea is that this phospholipid bilayer structure will form spontaneously. Very important to remember when we get to Unit 7. We've just talked about phospholipids, which form the basic framework of the membrane. Here they are, phospholipids, right over here. But there are many other important components of cell membranes. Proteins, cholesterol, and carbohydrates play key roles in membrane structure and function. Proteins play the following roles. They're where the cytoskeleton can attach to. They can act as channels over here. They can act as receptors, like that's probably what this is over here. They can act as membrane-embedded enzymes that are performing enzymatic functions, but they're performing it as a part of the membrane. Cholesterol is this molecule over here. It's a lipid. It's composed of a couple of rings that are connected together. And cholesterol plays the role of membrane stabilization. It's kind of a fluidity buffer. And it plays a double role. When it gets too cold, cholesterol keeps the membrane fluid. When it gets too warm, the cholesterol keeps the membrane's phospholipids from flying apart. And there are glycoproteins. That's a big word, but that's a protein that's connected to a short polysaccharide. I didn't write this out, but you also see the term oligosaccharide. That's a short saccharide. So those are involved in cell recognition, among other things. So like, for example, if you've ever heard of blood types A, B, or O, that's all about glycoproteins that are on the surface of red blood cells. And when we get to Unit 5, when we talk about the inheritance of blood type, we'll talk more about that. The membrane is described as a fluid mosaic. And what that means is that you have phospholipids and proteins and cholesterol that are in motion. So it's fluid because the components are moving laterally. They're not fixed and locked in place. Everything is moving around. You should think of like a droplet of oil that's floating in water, and think about how dynamic that is. All of those little fat molecules in that droplet would be moving. Well, it's true of the membrane as well. It's a mosaic because it's composed of a variety of pieces. And here is a mosaic piece of artwork, and it's composed of different tiles, and those tiles are put together to create the art. So within the bilayer, you have phospholipids, proteins, and cholesterol. And then inside and outside the bilayer, you have additional molecules that might be attached to proteins or the phospholipids. And that includes glycoproteins and glycolipids. A glycolipid is this over here. So a glycolipid, you can see this phospholipid tail. But what we've done is we've replaced the top part of it with a short polysaccharide, so glycolipid. And I misspoke. I didn't mean a phospholipid tail. I meant two fatty acid tails that are then connected to a short polysaccharide. So that's what we've got going on in terms of the fluid mosaic model. And here's a little animation of that.
On a cell membrane's not just a phospholipid scene. There's cholesterol, carbohydrates, lots of proteins. In fact, membrane proteins have a presence so great they often exceed the phospholipids by weight. And all of these components are in constant motion, moving, mixing like a boiling potion, flowing like dancers in a party in Passaic. That's why the membrane's thought of as a fluid mosaic.
As a fluid mosaic. And a little shout out to those folks who might be watching me in Passaic, New Jersey. I hope you're doing well tonight, and I hope you enjoyed the fact that not much rhymes with mosaic other than Passaic. Lucky for you. Let's talk about how proteins fit into the cell membrane. The terms I'm going to introduce right now are not important in and of themselves, but it is quite important that you understand the basic mechanism that we're discussing here. So we've got transmembrane proteins. Transmembrane proteins have a hydrophobic core that fits into the nonpolar inner portion of the membrane. So here's where you have to use the stuff from Unit 1 to help you understand Unit 2 if you're an AP Biology student. And a more general way to say that is use the biochemistry that you learned at the start of the course to understand the cell biology that you're learning right now. Here's a protein. Proteins are made of amino acids. These amino acids over here would have hydrophobic R groups that would be sticking out and, in a way, dissolving into this hydrophobic portion of the membrane. But these amino acids over here and down here would have hydrophilic R groups, and that would enable them to interact with water. So that's how a transmembrane protein works. And that's exactly what I just said in that text right over there. An integral protein is this shown at number 5. And again, these terms are a little bit overlapping. Integral proteins are built into the membrane. And in this case, this part of the protein would again have to be hydrophobic. The amino acids would have hydrophobic R groups, where these would be hydrophilic R groups over here that would be interacting with the watery cytoplasm. And a single hydrophilic region juts into the cytoplasm or the cell exterior. And then finally, you have peripheral proteins like number 3 over here. And those are just attaching to the phospholipid heads that are on the cytoplasmic side of the membrane or the cell exterior, which means that these amino acids over here would have to be polar, interacting with these polar heads of the phospholipids. Again, the terms are not important, but if you can explain how a protein like this could fit into the membrane, what amino acids you'd expect to find over here, you'll do well, because that's the kind of thing that the College Board and specifically biology teachers would like to put on your next unit test for Unit 2 of AP Biology, or your cells test if you're in an IB or a college biology class. If you're watching this on replay, just feel free to leave a comment, and I read my comments with enthusiasm, and I'll respond to it. So please leave a question right now if you can, and if you can't, then leave one later when you're watching on replay. Let's move on to the second part of the lesson, which is about membrane transport. Diffusion is how many substances enter and leave cells. Diffusion is a physical process that, is exploited by cells for biological purposes. So diffusion is about the tendency of molecules to spread out from where they're more concentrated to where they're less concentrated, And here's an example showing how this is a physical and not a biological process. If you put a drop of ink into a cup of water, well, that ink will start out as concentrated, but over time, it'll wind up diffusing until it's equally distributed. Why does this happen? These molecules are in random motion. So randomly, their tendency is going to be to spread out from where they are to where they aren't. They'll fill the container until the concentration is equal. A way to think about that is to think about molecules spontaneously flowing down their concentration gradients. So again, here's this drop of ink. It's spreading out. It was concentrated over here. It's diffuse over here. That's what diffusion is all about. It's about making things diffuse, moving them from concentrated to diffuse. And here's a diagram that shows this. You can see that these oxygen molecules are highly concentrated over here. Well, what they'll do is they'll spread out, and you can imagine them flowing down a concentration gradient. It's like a ball rolling down a hill. You don't need to add any extra energy. The energy is already there in the kinetic energy of the diffusing molecules. Diffusion is known as passive transport. Things are going from more concentrated to less concentrated. No cellular energy is required. And there are two forms of passive transport that you should know about. The first is simple diffusion. Simple diffusion is movement through the phospholipid bilayer. The other kind is facilitated diffusion, and that's diffusion through a protein channel. Let's talk about simple diffusion first. In simple diffusion, substances can diffuse across the phospholipid bilayer. What's important to know, and you might have to know this just by brute force memory if you can't reason it through, but small non-polar molecules like oxygen and carbon dioxide, they can diffuse right through these phospholipids over here and make it from one side of the membrane to the other. So I showed oxygen diffusing into the cell, and I'm also showing carbon dioxide diffusing into the cell. But really a much more realistic way to have shown this would have been oxygen diffusing in, because that's what happens as we do respiration. And then as we build up carbon dioxide, the carbon dioxide diffuses out. I'll change that the next time I rework this diagram. Lipids, such as steroids and fats, can also diffuse right through the phospholipid bilayer. So here's some kind of steroid. It might be a steroid hormone like testosterone or estrogen, the masculinizing and feminizing hormone that have such a big effect on our bodies as we develop. Well, they are lipids. They're non-polar. They can diffuse right through the phospholipid bilayer, and fats can do the same. These look like triglycerides, and they can make it right through the phospholipid bilayer. That's simple diffusion. Facilitated diffusion is for polar and ionic substances. So here's glucose. Glucose is going to require a channel. Why? Because glucose has all these hydroxyl functional groups on the outside. It's not going to be able to make it through. The cell needs glucose, and the way that the cell gets its glucose is by letting it diffuse through from higher concentration to lower concentration through a protein channel. Here's a channel for amino acids. Why can't they make their way through? Well, because they have a positively charged part over here. That's the amine over here. And here's a carboxyl group. So they're also going to need a channel to make it through the membrane. Facilitated diffusion. And finally, we have some calcium ions. How are they going to make it through? Again, through a channel. The non-polar phospholipid bilayer is never going to let something like an ion make it through. Let's talk about this word. If you speak Spanish, you know that easy is fácil. If you speak French, you know that easy is fácil. I probably pronounced those wrong just now, but you get the idea. Fácil, that's easy. And to facilitate is to make something easier. So these channels are facilitating diffusion of these substances. Again, here's the text that describes what I just said. Requires protein channels, transmembrane proteins that only let specific molecules or ions pass through. Those were both forms of diffusion, but sometimes cells need to move things from lower concentration to higher concentration. And to do that, they carry out a form of transport that's called active transport. And that involves pumping a molecule up its concentration gradient. So here we have a molecule that's in low concentration. In the cell, it's in higher concentration, but the cell still needs it. It can't rely on diffusion. So what will it do? It'll use energy to pump that molecule, that ion, from low concentration to higher concentration. That takes energy. You're going up a concentration gradient. When you go up a hill, it requires energy. You can't just slide down the hill, and that's what's going on here. Requires energy on the part of the cell. That energy is usually in the form of ATP to ADP, that transformation that we'll talk about so much in Unit 3 that's going to power the pumping process. Another way that can happen, however, is through the flow of electrons, and that happens in proton pumps that are used by chloroplasts and mitochondria to power ATP synthesis. That's a process that's called chemiosmosis. It is mind-blowingly cool. I cannot wait to teach you about chemiosmosis and that process of active transport. We're going to end this discussion of transport by talking about bulk transport. And there are two forms to know about: endocytosis and exocytosis. Bulk transport, what that means is that we're not just talking about molecules or ions. We're talking about large quantities of things. We're transporting things in bulk. With endocytosis, things are going into the cell. And note that endo, if you can sort of think about that as into the cell, then you've got it. Endocytosis into the cell. Exocytosis, if you think of the word export, well, export is when we send stuff out of the country. So exocytosis is about exporting. So we've got endocytosis and exocytosis. Those are both forms of cell transport. Endocytosis can have a couple of different forms. With large particles, it's called phagocytosis. When the cell is taking a sip of the extracellular fluid and thereby capturing some molecules, that's called pinocytosis. And then there's receptor-mediated endocytosis, where a molecule has to bind with a receptor, and that causes the formation of a vesicle. This is fairly advanced stuff. It probably will show up in a college course. It might or might not show up in an AP bio class, and you should consult with your course outline to see what's going to be on, the test that you'll be taking at the end of this unit about cells. Here's the basic idea. The membrane pinches in to surround a molecule, a particle, or some extracellular fluid, creating a cavity that becomes a vesicle. What's not shown here is that this would involve the cytoskeleton, and of course, this is a phospholipid bilayer. It's very dynamic, it's changing shape. Phagocytosis is really cool because it's used by single-celled eukaryotes to engulf and prey on other things. Here what we have is a single-celled eukaryote called an amoeba that meets a bacterium. It extends extensions of its membrane that are called pseudopods. That means false feet. It'll eventually surround that cell and then engulf it in a food vacuole. That food vacuole will fuse with a lysosome. We learned last week that lysosomes are full of hydrolytic enzymes, and it'll digest it into particles that can then be incorporated into the cell, and what can't be digested will be released through exocytosis. Your white blood cells do the same thing as they scour your body looking for invading bacteria or viruses. Receptor-mediated endocytosis has been exploited by viruses as a way to slip into our cells. And of recent memory, the SARS-CoV-2 epidemic is all about the SARS-CoV-2 virus evolving an exterior protein, it's called the spike protein, that was capable of binding with the ACE2 enzyme/receptor that's on the surface of kinds of tissues that are called endothelial tissues. They're tissues that, for example, line the surface of our lungs or blood vessels. Once that binding happens, that induces the cell to create a vesicle and swallow up the virus. So the virus tricks the cell to letting the cell invite the virus in. HIV does a similar thing. It binds with a receptor on the surface of helper T cells. It's called the CD4 receptor and does the same thing. It leads to fusion, and then the virus enters, has its RNA enter the cell and ultimately become DNA and then integrate into the nucleus. Viruses are really cool. We're going to talk about them in Unit 6 of AP Biology, so just keep on watching the AP Bio Show. And I've also got great tutorials about that on learn-biology.com because, you know, viruses are everywhere. They're important. People are talking about the Mpox virus a lot. If you want to learn how viruses work, that's a place where you can do it. Exocytosis is about vesicles that fuse with the membrane and then they release their contents. Speaking of the immune system, which we were just talking about, cells that are scavenging, swallowing up bacteria and viruses, when you get an infection, you have cells that are called B cells, and those B cells produce antibodies. How do they release those antibodies out into the body fluids, the blood and the lymph? They do that through exocytosis. The vesicles aren't shown here, but something to think about since you are now starting to know a lot of biology is that these proteins would be made in the rough ER. They would be sent from the rough ER to the smooth ER, Golgi. They'd be packaged in a vesicle. All of those vesicles filled with these antibodies, these proteins that are capable of binding with and neutralizing viruses, and then they'll be released from the cell. Biology is really cool. The immune system isn't a part of the AP Bio curriculum anymore, but if you want to dig deeper, I have a really great tutorial about that on learn-biology.com. Finally, we're going to talk about membrane potential. Membrane potential is an electrical charge across the membrane. It's created by cells. It takes energy to create it by pumping ions across the membrane. That's active transport. And a key example, and this is a preview of Unit 3, is that mitochondria will use a flow of electrons to pump protons from the mitochondrial matrix, this space over here, into this area over here, the intermembrane space. And that gives this intermembrane space a very positive charge. The matrix develops a negative charge, and that creates a force, an electrical force that will pull these protons through this channel over here. It's called ATP synthase. And as they do, as you can tell by its name, ATP synthase will use the kinetic energy of diffusing protons to make ATP. If that was like, whoo, Mr. W, that was a lot of information, that's cool. We're going to talk about that when we get to Unit 3. That's all about the electron transport chain. It is mind-blowingly fantastic stuff. I cannot wait to teach you about that. Oh, and here's a little bit more about membrane potential. There's another thing to know about. It's called the sodium-potassium pump. And my ability to talk right now, your ability to listen and think, is all about a membrane potential that's created across the axons of nerve cell membrane. Those are long extensions that extend along nerve cells. And here's a little animation. When you think a thought, it's partly because of waves of ions that are traveling down the length of a neuron, a nerve cell. Those ions can only travel because they were pumped there. So watch. All right, so that's basically what happens. That difference of ions on the outside, positive ions on the outside, negative ions on the inside, that has to be created. It takes a lot of energy to do that, and that is what membrane potential is all about. Well, if you want to learn more, I've got tutorials about all of this material on learn-biology.com. The tutorials, in this case, wouldn't be about cell size and cellular compartmentalization. They would be about membrane structure and function. I want to emphasize that the best way that you can set yourself up for success in biology is by going to learn-biology.com, which will give you the skills and confidence to ace your biology class and the AP Bio exam. It's 100% aligned with the College Board's standards. It's approved by the College Board for use as a textbook. It's really focused. It has interactive tutorials that'll teach you everything you need to know about biology. It's got readings, videos, flashcards, quizzes that teach you everything you need to know about every topic in AP Biology. The interactions include multiple choice questions, click and drag diagrams, identify the part questions, and free response questions that are going to get you ready for the AP Bio exam. You get positive feedback for correct responses and hints and repetition for incorrect responses. You will learn more than you'll ever believe. We have student learning guides for every topic that guide you literally through the tutorials that tell you exactly what to do and everything that you need to get ready for the AP Bio exam. If you're a teacher, we've got student performance monitoring, and free trials are available for teachers and students. Go to learn-biology.com to learn more and to sign up. Sickle cell disease. This is Sickle Cell Disease Awareness Month. And what is sickle cell disease? The reason why sickle cell is important to know about as a biology student is that it hits on practically every unit in the course. And in addition, just to be an educated person, you should know about this incredibly important and destructive genetic disease that is now on the cusp of being, I don't want to say cured, but being able to be effectively treated because of new advances in the greatest subject of all, biology. So sickle cell disease is an inherited disease that affects hemoglobin in red blood cells. It affects 100,000 US citizens, mostly African Americans, and about 8 million people worldwide. The symptoms, the complications include pretty much every part of the body. So for example, if you have sickle cell disease, if you've inherited this disease, there are central nervous system problems such as stroke. And most of these problems are related to clotting and inflammation that's caused by this disease, improper blood clotting and very large degrees of inflammation. You can have problems related to the heart, again, caused by blood clots that can get into the heart and the lungs. These blood clots can affect the kidneys. They can affect the liver, leading to this production of a substance called bilirubin, and that causes yellowing of the whites of the eyes. But that's really just a symptom that shows you that there's some underlying stuff that's not going on. There's some underlying stuff that's going on. And then, most importantly, there are acute, widespread, chronic pain crises, and the pain can be debilitating. Now, over the course of my experience as a biology teacher, I first taught in 1987. That's the first time I was in a classroom, long before many of you watchers were even alive. Well, at that point, sickle cell disease meant early childhood mortality. But now people with sickle cell through medical treatment are living to 50 years of age and even longer. And there are cures related to biotechnology and genetic engineering that are on the horizon. The inheritance of sickle cell is autosomal and recessive. What that means is this. We'll talk about this when we get to Unit 5, but autosomal means that it's not on the X chromosome. Recessive means that you have to inherit the disease from both parents. So you have to inherit the gene from both your parents, and there are a couple of possibilities. You can be big S, big S. That means you'd have two normal alleles. An allele is a gene variant, and that means that you don't have the allele at all. So you're what's called homozygous normal. Both your chromosomes that carry this gene don't have the defective defective gene on it. You can be heterozygous, where you have one allele. And I wrote over here, no symptoms. You can have slight symptoms under some very extraordinary circumstances. And there is one big effect that we'll talk about a little bit later. So hold that question in your mind, like what can things like being a heterozygote, what can that affect? And then if you're a little s over here, then you're affected. So in a typical mating between two people who are heterozygotes, big S, little s, you could expect 1 out of 4 of their children to be affected. Okay, sickle cell anemia is caused by a point mutation in the gene for hemoglobin. Hemoglobin is a quaternary protein. In other words, not just a tertiary protein that's folded up into a specific shape, but you have multiple polypeptides that are interacting. And in this case, there are 4. There's an alpha chain, and there are 2 of those that are 141 amino acids. There's a beta chain, and there are 2 of those that are 146 amino acids. So we've got 2 alpha chains, 2 beta chains. And a point mutation causes a change of this, which is what people who don't have the disease at all have. They would have CTC. If you have the mutated hemoglobin, you have CAC. So what's phenomenal is that this entire disease is caused by a single mutation, a substitution of A, adenine, for thymine. Now, why is that a big deal? Because DNA makes RNA makes protein. And this mutation changes the amino acid. Instead of glutamic acid, which is an acid, we have valine, which is non-polar. So that's the molecular cause of sickle cell anemia. Now, what's the big deal? You have this valine instead of glutamic acid. It's a big deal because if you have valine, then what happens is that these mutated hemoglobins will have that valine sticking on the outside. And those valine molecules will cause hemoglobin to interact with one another, causing fibers. Normally, hemoglobin molecules are quite individualistic, and they won't have anything to do with the other hemoglobin molecules in a red blood cell. But if the person who's making these red blood cells has the defective form of the gene, then what will happen is those hemoglobin molecules will interact and they'll form these fibers. It's like a, not primary, not secondary, not tertiary, not quaternary, but it's like a 5th level structure that emerges. And those fibers will cause the cells to sickle. They'll become spiky instead of smooth. And those spiky cells tend to get caught in the smallest blood vessels, which are called capillaries. It's all about sickling, leading to clotting and inflammation. The inflammation is caused by the fact that these kind of spiky areas of the blood cell, they're prone to membrane rupture, which releases hemoglobin directly into the blood as opposed to keeping it inside the red blood cells. And that can lead to inflammation. So that's what sickle cell does, and that's what it causes. And now we see how it can cause sickling and inflammation. So if this disease is so bad, why is this allele so common? Why are there millions of people around the world and hundreds of thousands of people, or 100,000 people in the United States, who have it? An allele, as I said before, it's a gene variant. And so we've got two variants. We've got HbA, that's normal hemoglobin, and we've got HbS, that's sickle cell hemoglobin. In Nigeria, the country in the world with the highest level of sickle cell disease, 2% of all babies are born with the disease. Between 20 and 30% of the population are heterozygotes. In other words, they have one normal gene and one sickle cell. Why would these rates be so high for this deadly disease? The frequency of the allele in the gene pool is between 10 and 15%. The answer is because even though sickle cell disease is a dreadful disease with all these terrible symptoms, that sickle cell allele in heterozygotes disrupts the life cycle of a parasite that causes a tropical disease called malaria. Malaria is caused by a eukaryotic protist that infects red blood cells. So think about that, a cell that's so small that it can get inside other cells. It's not a bacterium though, it's a eukaryote. And the parasite is spread by mosquitoes. It causes debilitating fever. And listen to this. This disease affects 249 people and caused over 600,000 deaths in 2022. So malaria is a dreadful disease. If you have one copy of the sickle cell allele, in other words, you're not little s, but you're big S, little s, you're a heterozygote, you have resistance to malaria. And that's why there's this strong correlation between the frequency of the sickle cell allele and the incidence of malaria. Dark red over here means more sickle cell allele. Dark green over here means more malaria. That's why this evolved. It's called heterozygote advantage. Now, this is something that we'll talk about in detail when we get to Unit 7, but it's fantastic to know about it now. If you are a heterozygote, big S, little s, you're protected from malaria. If you're little s, well, then you have sickle cell disease, and that's a tragedy. And if you are big S, like this person over here, then you can get malaria. So it's an evolutionary trade-off. There's the cost of the disease versus the benefit to these folks, which have a high level of resistance to malaria. That's what it's all about. Now, just to end on a hopeful note, there are new gene therapies. There's already medical treatment that's available for sickle cell that vastly extends lifespan. And that's especially true in countries like the United States, where even though there are many health inequities, I don't want to pretend that there aren't, but despite the fact that there are health inequities, medical care is going to be better than in a place like Nigeria in general. So what has to happen? Well, you have cells in your bone marrow that create red blood cells. And the gene therapy treatment involves this. You would remove stem cells from a patient who has sickle cell anemia. So you'd extract them. It's not an easy procedure. And then you'd use chemotherapy to eliminate all the rest of their red blood cell, all their stem cells in their bone marrow. That's a painful procedure. You're very immunosuppressed when that happens, but that is the first two steps. Now, you'd use a technique called CRISPR. That's a gene editing technique that we'll talk about in Unit 6 that can find and deactivate that sickle cell DNA in the stem cells that have been extracted. So all of that bad gene is going to be knocked out. And what you can do is you can use a virus to introduce the correct DNA. So once you've done that, you can infuse those corrected cells, right? So we've replaced the broken allele with a good allele, and we're going to give that back to the person. They'll colonize the person's bone marrow, and no more sickle cell anemia. that person would be cured for life. Now, that is one way of curing the disease. And another way is reactivating what's called fetal hemoglobin. We have two kinds of hemoglobin. When we're fetuses, we produce what's called fetal hemoglobin. It's got 2 alpha chains and 2 gamma chains. And what that fetal hemoglobin can do is it can actually pull oxygen from the mother's bloodstream and send it into the fetus. So in this other form of gene therapy, what you do is you reactivate the gene for fetal hemoglobin by deactivating this gene that's called BCL11A. And you'd use CRISPR and other gene editing techniques to deactivate BCL11A, and that would allow a person to start producing fetal hemoglobin. And if you can produce even a small amount of fetal hemoglobin, it ameliorates most of the symptoms of sickle cell. So these are new gene therapies that are coming down the pike. Again, we'll talk more about this in Unit 6. Biology, amazing on every level, both in terms of understanding the evolution and in terms of the future that we're all looking at. Let's take our weekly quiz. And to do our weekly quiz, we are going to go here. You're on the main page, and then you just go to AP Bio Unit 2, and then you go to the quiz page. That's at the end. It has there, we've got a review page. So here we are, and we're going to start the quiz, and I'm already logged in. That's good. And you can pick the questions that you get to do. So we just did membranes, plasma membranes, membrane permeability, membrane transport. So that's just 6 questions. I don't think we'll get through all of them, but we'll get through some. So what I want you to do is this. First of all, okay, in the diagram below, which membrane component? This is a diagram showing the phospholipid bilayer, and A is pointing to some carbohydrates that's sticking out of molecules that are on the outside of the membrane. B is talking to a molecule of cholesterol. E is pointing to an integral protein. D is pointing to some phospholipid heads, and C is pointing to a channel. So in the diagram below, which membrane component is responsible for maintaining an optimum level of membrane fluidity? I want to see a letter. I want to see it in the chat. Go ahead and do it. Show me the answer. Let's show me the answer right now. Want to see that quiz? Do your best. Just put the answer in. Nothing bad can happen. Just want to encourage you. I want to see a letter, A, B, and what do we got? We've got one answer so far. Any other answers? We've got more people than that who are watching the show. Any other answers? And we're going to go with that answer. Let's see what happens if we click on B. Fabulous. B is pointing to cholesterol, which acts to maintain the proper level of membrane fluidity. Fantastic. Oh. In the diagram below, which membrane component would be involved in cell recognition? It's the same diagram that I showed before. A is pointing to some carbohydrates sticking out from the membrane. B is pointing to cholesterol. E, an integral protein. D, phospholipid heads. And C is a protein channel. So involved in cell recognition. Anybody? Let's see an answer. Go ahead, be courageous. Put in the answer A, B, C, D, and E. And the answer, let's try A. Nice. A is pointing to short polysaccharides, oligosaccharides that are attached to lipids or proteins. Oligosaccharides are involved in cell recognition. C is a channel. So it's not really involved in cell recognition, just responding to one of the answers. All right. So next question. Everybody ready? Let's go. Okay. The diagram below illustrates a process related to glucose homeostasis in liver cells. For those of you who are in the listening audience, there is insulin with an insulin receptor. That insulin receptor then sets off a phosphorylation cascade. And then there's some structure inside the cell that's producing vesicles with a GLUT4 vesicle that's allowing glucose, with a protein that's allowing glucose to enter into the cell. So you're focusing on this part of the diagram over here, and there's a lot of glucose outside, there's only a little bit of glucose inside. So the question is, glucose is entering the cells by, first choice, let's call that A, simple diffusion. Second choice, receptor-mediated endocytosis. Third choice, active transport. Fourth choice, facilitated diffusion. Do we have an answer? What do we got? A, B, C, or D? Simple diffusion for A, receptor-mediated endocytosis for B, active transport for C, or facilitated diffusion for D. Let's see a letter in the comment thread. I can't wait to see that letter. Let's see it come in. Don't be afraid. You'll just do your best. You got this. Let's see it. Any choices? A, B, C, or D? And we've got over here, you can see that the glucose is in high concentration outside, low concentration inside. So that's got to be facilitated diffusion. Why? Because it's coming through a protein channel. Okay, so that's why facilitated diffusion. Let's check that answer. Fabulous. This was facilitated diffusion, which involves diffusion through a protein channel, as illustrated in number 2 below. We're going to do one more question. A scientist has constructed an artificial cell membrane that consists solely of a phospholipid bilayer. A portion of this membrane is shown below. For those of you who are listening, all that we're seeing is a phospholipid bilayer. Which of the following substances would diffuse most rapidly through this artificial membrane? First choice, A, glycogen, a polysaccharide. Second choice, glycine, an amino acid. Third choice, fructose, a monosaccharide. Fourth choice, calcium ions. And fifth choice, oxygen. So let's go. A is glycogen, B is glycine, C is fructose, D is calcium, and E is oxygen. What do we got here? Which of these molecules could make it through this phospholipid bilayer? Let's see an answer into the chat. And I'm seeing E, oxygen. Do we have any other answers coming in? Okay, well, let's do this. I'm going to choose something that I know is wrong. I'm going to say glycine, an amino acid, and let's see what happens. What our quizzes do is they'll tell you that molecules with charges, like the amino acid glycine, can't diffuse through a phospholipid bilayer. Next time, look for a substance that's small and nonpolar. And those of you who are thinking oxygen, you were correct. Fabulous job. This is great. Hey, I'm noticing that it's 7:33. We're past our hour. I am really grateful to those of you who are still watching the show. Let's go back to our slideshow. And let's talk about your plan for this week. Your plan for this week is to go to learn-biology.com, then AP Bio Unit 2, and then cell structure and function. So we've got tutorials about membranes that you can do on learn-biology.com. I don't think I see any questions that are in the chat. So really what I want to say is thank you. Thank you so much for joining me. Thank you so much for spending your time with me tonight. If you're enjoying the show, then really the best thing that you can do is tell a friend to watch. And yourself, go to learn-biology.com. You got a free trial, 2 weeks, no risk, no cost. You will learn more than you ever believed, and you will have a fantastic time. So I am really happy to have taught you tonight. I'll see you a week from tonight. Have a great week, and please watch this next video. Those of you who are watching on replay, the next video that's coming up, really appreciate you having been here. Really looking forward to learning more biology with you. Thank you so much.